participants yesterday presentation vegetable seed production, they were asking that any tools to reduce the cost. So everybody is very much interested in reducing the cost of this seed production, and especially for the hybrid seed production. Yes, seed production is a conclusive phase of breeding activity. Unless you throw your seeds to the society, there's no the point or the importance for you. Your importance will be known only when your variety seeds are available for the end users, that is to the farmers. That's why it is very, very important sector of this agriculture production. And of course, the seed production, definitely it is important as the end product of the breeding activity, but more so hybrid seed productions. So hybrid seed production is more important because the hybrids are very important. Hybrids are far superior to the non-hybrid varieties of the same variety. This is very, very ways it depends on whether you the OP variety is the similar potentiality of the hybrid or not. It depends on the genetics or the character or that yield in that particular crop. It depends if it is because for dominance, if the hybrids are superior, then it is not possible for you to develop varieties and make your seed production very easy. So, but most of the cases it is dominance, maybe the epistatis is involved. That's why, of course, epistatis also it is difficult to exploit from the OP varieties. It's only additive component. If it is more, definitely your OP varieties are better than the hybrids. The dominant part, of course, you can exploit it by OP varieties, no doubt in that. But it is quite the true in some of the crops where ester years, the ester years hybrids, whatever the potential it was there, it is staying there for the OP varieties also. So that is one important thing. That's why it is inevitable for us to get into this hybrid seed production, which is very cumbersome. Yes, they are superior. The hybrids do not breed true. This is one more, the characteristics of these hybrids. That's why they are natural patents. That means nobody can take your technology. But this is sometimes from the private seed companies or exploited. That is the other threat. But yes, if you want to get the credit for this, every time farmers have to come to you and to get the seeds. That's why it's a kind of patent. You are protecting your technology. So that's why the most of the private seed companies are into this hybrid business, and our production has tremendously increased because of the hybrids. We are able to feed this increased population because of the hybrids of field crops, definitely. Then the cost of hybrid seed production is greater than that of the open pollinated seed production. So that is every time you have to cross the parents and get the seeds. That is, OP varieties are just this, the crop to naturally pollinate and just collect the seeds by maintaining certain standards of isolation and also field standards. That's quite easy compared to the hybrid seed production. The hybrids dominate the commercial vegetable market because the heterozygous is strongly expressed in very many instances. That's why they are there and we are able to exploit them. So because of the lack of uniformity in okay, one more thing is hybrid seed production that will get you the uniformity, and that's why one more reason that you get into that. But the hybrid seed production is many times maintenance of parents is hindered by the inbreeding the inbreeding depression in some of the crops, especially coal crops. You have a problem, right? So in that sense, what to do to overcome this problem? So people have resorted for going for this double cross or triple cross or three-way cross hybrids. So if you get into that particular kind of exploitation of a hybrid bigger, then you are going to lose the uniformity. That we have to bear in mind. But it is practically true in some of the coal crops where not the single cross are popular commercially. There are double cross and even the triple cross, three-way cross hybrids are popular. So hybrid seed production is cumbersome, expensive, and requires skill and technique. So it offers the employment also, many of the people, right? To many people, involved. It offers you more people to work and earn. So emasculation and hand pollination, these techniques are followed in the crops where it is feasible, but it is not the case in all the crops. But yes, even hand pollination and hand emasculation, although it gets you a lot of employment opportunity, but some of the developed countries, and even in India also, we are going to face the problem of labor availability. So yes, up to now we are following it. In future, it may change. The hand pollination and hand emasculation techniques are followed in eggplant, sweet pepper, tomatoes, and okra, which is very cumbersome and which enhances the seed cost. It's not only the cumbersome and complexity and all that. Human error, anywhere it goes, you have a problem in your hand. That means many seed companies, they face this problem when they give the F1 hybrids for commercial cultivation. So besides, the cost of the hybrids is higher because all this work is involved and it is going to cost you more. So here the genetic tools can aid. That means they can help in reducing the cost of seed production. So you can make this hybrid seed production economically viable. It is not possible to use that the systems like hand pollination and hand emasculation in all the crops unless you have these genetic tools being used. You cannot have the hybrids. So that's what some of the field crops, they have got these systems, and they are able to produce the hybrid seeds and market it, and the total production has increased by that. The three commonly employed genetic tools for overcoming the emasculation and hand pollination, these are the two important activities in hybrid seed production. So those these activities can be overcome, or you can have the alternatives by using some of the genetic tools. Three important tools are sex expression in especially vegetable crops, specifically the cucurbits, and the self-incompatibility in crucifers or cole crops, and of course male sterility across the vegetable crops. It is being used. And of course, the genetic tools, if you say that there is a chance of instability of these systems, and if something happens and it is giving you the mixture of the seeds, there you have to use the markers. So there is a one more horizon of this genetic tools being used in hybrid seed production. By use of the markers, you can overcome those problems of selfing, right? So that's why the genetic tools are important. So methods of hybrid seed production using genetic tools, if you just look at here, the important the things which we are using. First one is the use of sex expression. There the monoecious lines, so pinching of staminate flowers plus manual or natural pollination. Sometimes you cannot completely overcome this pollination. Sometimes you have to do it, or you have to take the help of insects, or by even human intervention also is required in some of the crops to increase the seed yield of the F1s. So this is being followed in bitter gourd and bottle gourd, and Gynoecious lines plus natural pollination, it is being followed in cucumber, musk melon. And the Diocious species also, sex expression, it's manual or wind pollination. It is possible in Diocious species like spinach, asparagus, and sweet corn. So this can be used for the hybrid seed production. The second important thing is that use of male sterility. So male sterility plus manual pollination, you can do it. Manual pollination, it can be followed in case of tomato, hot pepper, sweet peppers. Especially the pollination, manual pollination is required in highly self-pollinated crops like tomato, because the pollen transfer mechanism is not that efficient if you avoid the selfing. What you do it in hybrid seed production, you are crossing the parents. There you can avoid by male sterility, but the pollination has to be done to increase the seed yield. And also possible male sterility plus natural pollination, that is possible in case of onion, even cole crops, although you have got self-incompatibility. People are looking for this male sterility in those crops: cabbage, cauliflower, carrot, radish, musk melon, right? So the maintenance of male sterility it depends upon the system type of male sterility being used that we can just discuss. And then the use of self-incompatibility is the one more genetic tool, that is self-incompatibility plus natural pollination, that's being followed in cole crops like cabbage and cauliflowers. And in addition to these three genetic tools, to eliminate the chance of selfing, the seedling markers are used. The markers are used. The markers can be of morphological or molecular markers also can be used to identify these self seeds. So just look at the methods of economic production of hybrid seeds using genetic tools. The easy, the emasculation of a female parent and the effective pollen disposal are the two important activities required for hybrid seed production. The genetic male sterility, which is male sterility, I said it again, they have got different types of male sterility systems. Genetic male sterility, it avoids emasculation. It can be used in tomato, muskmelon, and squash where it is reported. So expression of male sterility is influenced by the temperature. That's the one problem that always you have to look at. It is simple that you have got a maintainer line, the B line, which is in heterozygous condition, and your A line, that is a homozygous recessive for male sterility, that gives you the male sterility. And always you cross these two and you collect the seeds from the A line, and you will get always 50% segregation of heterozygous and homozygous recessive. That means 50% population will be male sterile and another 50% will be that will be a maintainer line. So it goes on like you can maintain. But at the time of seed production, you are going to use the C line. There is another combiner, that one more, the parent, the female parent and then male parent. That is one more combination of the parent where you are getting it identified through your combining ability and heterosis studies. That is C line, but the C line has to have the homozygous dominant gene for the male fertility, right? The male fertility. So that you got F1, which is heterozygous, and no problem as far as reproductive organ is concerned or your economic part is concerned. It can be maintained under heterozygous condition by back crossing with the recessive parent. And this is quite, as I explained, this can be done. So for yes, the commercial hybrid seed production, heterozygous seed stock is grown, which segregates into 50% heterozygous male fertile and 50% homozygous male sterile. So male sterile plants can be identified at early stage. This is the problem here in use of genetic male sterility. So 50% of these plants you have to identify before they flower. If you identify them after flowering and after pollination, so it's a waste that is contributing the pollen, and it is going to spoil your entire seed production program. So that means if you identify this 50% of the population which is male fertile, and you had to remove them, okay, you have to remove them. So identification, you have to use some markers, recessive markers, always going to be useful, which is linked with the male sterility. You have to, otherwise if you wait till the flower, the flowering occurs and you could see the flower and identify that this is fertile and sterile, by that time then you have to remove all the flowers already open till you identify that plant. And if you are delaying, then it is going to add to the problem. So that's why many times, unless you have got a markers here, many times the seed production activity has been, it is not successful. Then you have got the functional male sterility. This is being looked at in Solanaceous vegetable crops. Maintenance is easy. That means there is pollen fertility is there. Maybe the anther is not dehiscing, and maintenance is easy because you can just forcefully open the anthers and go for self-pollination. You can maintain the parents. But as such, anther is not opening. That means the pollen selfing is not happening. Then you can go for, but it has to be used with the markers only. Or even it is being you get because of the breakage of sterility, that little portion of self seeds will be there. To avoid that, you can use the marker, or if you want to tolerate that amount of selfing, it's all right. But it depends on the how successful that hybrid is. exerted stigma along with the marker with tomato. This is also, I'll tell you what has happened in tomato when we discuss on tomato. So next system is the cytoplasmic male sterility. It's onion where it is being exploited for hybrid production. Very long back it is being done, and in all the developed countries. You have got sterile cytoplasma gene and the normal cytoplasma that is required. A line is having the sterile cytoplasma, B line is having normal cytoplasma you have to search for. And every time you can produce the seeds of A line with this maintainer. But then when you want to go for the hybrid seed production, you have to have your second parent, that is a male parent C line, which is having the normal cytoplasma. So this type of male sterility is determined by, the cytoplasma, as you know. So it inherited from the female parent, and that's why every time you are getting the sterile progenies. So that's why, its maintenance is easy. So nuclear genome has no role to play, and it is identical to the female parent. That you have to ensure. So nuclear genes, maintainer line, it should be isogenic with the A line with respect to all the nuclear genes. So this has got a greater value for bulbs, because where the problem here is your hybrid will be sterile. Right, you are hybrid because the cytoplasma is inherited from the female parent. You are collecting the seeds from this A line. So the hybrid, what you get, it is going to be sterile. There you have got a problem. That if economic part is fruit or seed, this genetic tool cannot be used. So it is okay, quite successful in bulb crops where the bulbs are economic part, not your seed or fruit. And same is the ornamental crops. This mechanism can be used and being used. Then the next one is the cytoplasmic genic male sterility. Here the cytoplasmic male sterility, where nuclear gene for restoring the fertility is identified, is known. So that means the same cytoplasmic male sterility can be used for the other crops where the seed or fruit is economic part. So that's why this is a kind of extension of this cytoplasmic male sterility, cytoplasmic genic male sterility. The gene is referred as a restorer, which restores the fertility in F1 and by virtue of its nuclear location. So in cytoplasmic male sterility, when this restorer gene is known, then it becomes just cytoplasmic genic male sterility system. That's all. So this system is known in maize, jawar, bajra, sunflower, wheat, and of course among the vegetable crops, capsicum is the candidate, and that is even in chili, right? It's own same capsicum annum. So maintenance of male sterility, you have got A line and B line. So A line sterile cytoplasma, B line normal cytoplasma, but nuclear gene, it is identical to A line only, right? You have to develop it through a backcross breeding. So same, A and B lines are same except that cytoplasma gene for fertility. So that means every time you can produce the seeds of this A line, and when you go for hybrid seed production, you are planting the A line, and the C line is going to be the male parent, which should have a restorer gene. That means that for a male sterility, it should have a homozygous dominant male sterile genes. Then only your hybrid will be in heterozygous condition, which is fertile, and whether it is seed or a fruit, it is going to set. So that is the advantage extension. The production of using the CGMS system, you can just look at here the double cross hybrids. Also, you can produce seeds. So inbred A, CMS, inbred B, non-restorer, male fertile, and one side you are doing it. Another cross, second cross is inbred C. It is again CMS, and inbred D. It should have a restorer male fertile, right? Then you get this two single crosses, where first single cross is just like the sterile. It is sterile cytoplasma and sterile nuclear genes. Whereas second one is it is the single cross second, right? It is male fertile. Cytoplasma is sterile, right? But the nuclear gene is in heterozygous condition. It gets the fertility, and this single cross 1 and single cross 2 are grown together, and you are going to get the seeds, the double cross hybrid seeds. So A into B into C into D. So here, of course, some kind of uniformity you are going to lose. It is going to segregate here, right? It depends on the 4 lines you are going to use. Why it is being done? If you have got a parent seed production problem because of the inbreeding depression, this is being used, right? So, but here in the field, you have got the 50% of the plants will be double cross hybrid. They will be fertile and 50% will be sterile. So these 50% plants will supply the pollen for the other plants, and you may not have a problem for even if it is the seed or the fruit is the economic part in that crop. That is the maize sterility, and then some developments we can look at individual crop wise. The self-incompatibility system is one more genetic tool being used in coal crops. You have got a sporophytic kind of self-incompatibility which is being used to operated in these crops using by using this one can produce the single cross, double cross, triple cross, or three-way cross hybrid seeds. So you know this the self-incompatible means on selfing they do not produce the seeds, right? Because the pollen will not germinate onto the stigma, although it is a receptive. So at the stigmatic surface itself, it is identified that the pollen will not germinate of same allele. If it is having multiple allelic system is there, it will not germinate. But if you cross with another allele of this incompatibility, self-incompatibility, where multiple allelic system as it is being shown, you got line A for a single cross, S11 is it's a homozygous for self-incompatibility. Line B is S22. So these two alleles are different. These two alleles are different. That's why the seed set will be there. So quantity of hybrid seed production is low. And you can go for the double cross, three-way cross. You have to construct the lines of each line having a different self-incompatibility alleles, right? Because it is the same location. Same single gene is there, but multiple allelic system is operated. You have to have a different alleles. So that is being used in cross, three-way triple cross. Also, you can produce it. It is less uniform hybrids, no doubt. You have to accept this disadvantage because you are resolving the issue of inbreeding depression where the parental seed production and hybrid seed production is also low. You want to increase the seed yield because that has to be supplied to the farming community in a large quantity. It is the only way in some of those crops. Yes, self-incompatibility and also the male sterility. Now the next one is the Gynoecious, Monoecious and Gynoecious lines. The hybrid seed production by using these sex forms are being practiced in cucurbits. These forms are used production of hybrid seeds in cucurbits, especially in cucumber at commercial scale. So Gynoecious plant actually that produces Gynoecious and also Monoecious, and intermediate forms are there. The latter two are uprooted as soon as the first staminate flower appears, and remaining Gynoecious plants are left for hybrid seed production. You have to wait and see the flower and then you can do it. This is quite possible because the flowering pattern is such that first you will get the male flowers and then you will get the female, like that. The sequences are there in majority of these crops, and this is quite handy. You can go for. But yeah, Gynoecious lines you can maintain. You need to have the the Monoecious lines. Several generations of backcross you have to develop such that I'll discuss this in when you discuss on the cucumber. Gynoecious lines maintained by, of course, there is a other way also, other than the use of the genetic tools. Right, you can go for growth regulators. You can maintain these parental lines because, as it is advantageous, same time it is also disadvantageous because most of the time it is not stable. Slightly temperature increases, and then you will get the male flowers, and the day length also, which is going to influence. So the genetic system is being influenced by the environment. That is going to be sometimes disadvantageous. You may get some self seeds. There's a problem. So muskmelon is also being used here. You have got a hermaphrodite and monoecious first, and you have to go for several backcross generations, and then you have to construct this Gynoecious line of dominant A and recessive G, so that it will give you the Gynoecious line, and then the hermaphrodite, it is homozygous recessive. So exact genetic makeup of the Gynoecious in muskmelon is not correctly understood due to the temperature and the photoperiod effects. This is always there. It adds to the confusion. So the Gynoecious breeds true in short day and low temperature conditions. I mean, if you alter the temperature again, this genotype it behaves differently. Under long day and the high temperature condition, it becomes the Gynoecious. So it starts producing the male part, male flower. GA will not induce the maleness here. However, perfect flower production was reported with the MCEB, the chemical, at a true leaf stage. Sometimes they will not respond. That have to, develop, standardize for your own parent, right? Many times people will not be, revealing what's happening there. But yes, so they may not respond. You have to identify certain chemical which your line can respond specifically. These techniques can be extended to other cucurbits. What it is given the example, cucumber and muskmelon can be looked at other crops also, other cucurbitaceous vegetables. So Monoecious in spinach and Diocious in asparagus can be used for hybrid seed production, quite possible. Yeah. So now you just look at the history, this commercial hybrids in vegetable crops, during the first half of the 20th century, field corn, sweet corn, the years also it is given eggplant, summer squash, tomato, cucumber, onion, watermelon, winter squash, pepper. And during the third quarter of the 20th century, the muskmelon, cucumber, pickling types, again, they are the slicing types and pickling type come here. The cabbage, grain sorghum, sugar beet, broccoli, spinach, red beets, Brussels sprout, carrot, pearl millet, coconut, of course, cauliflower, alfalfa, barley, various other crops of the rice and sunflower also has come in. During the fourth quarter, you have got asparagus, celery, oats, rye, cotton, potato also, and soybean. So these, of course, the legume crops, it is difficult, but yes, you are getting that in highly impossible crops like legumes also. We are getting the hybrids. Okay, that's the hybrid story, but use of genetic markers only, I'll just concentrate here. The tomato, I think, I don't know how much time I may get it here, but at least few crops I'll discuss. So use of male sterility and hand pollination is said. 3 types of male sterility is known in tomato, and all of them are governed by a single recessive gene. So except one few cases I'm just mentioning here, series of MS genes are produced, they are going to produce the sterile pollen. All of these genes are recessive except MS48, which is dominant. To facilitate the elimination of 50% of the fertile plants, the morphological marker anthocyanin less, which is closely linked to the MS1035 gene, is used. So it depends on the strength of the linkage between the genes. Unless you have got very strong linkage, there is a breakdown, and then you are ending up with a problem. So because that is going to be indicated. So what is it? This, the certain papers also published, like auxotropes in other crops also. They are nutritional mutants. What is that actually? It is, you need not have to even identify and kill the plants. The plants carrying this anthocyanin less on selfing, if you are incorporating this into your female parent, on selfing, the plant will die on its own, right? Die on its own. And you can use it for hybrid seed production. That is being done in developed countries where the labor is very costly. But some breakage, that the strength of linkage, it depends on. Then you may end up with some self seeds, but still plant is not dying because the dominant allele is, if it is there, it's coming, then you have got a problem for self seeds in a hybrid seed lot. So this, how to maintain the parent, is also a question here. But for everything they have developed, you can apply the thiamine or some amino acid specifically for that particular parent. Where it is lacking, you have to spray that. You have to spray, and you can regenerate that plant. Parental seed production, you can do it, but this is not easy as it is said, but it is possible. So this is a series of MS. Even double tagging also we have seen. Maybe some of the companies are doing all this business. They may not publish it. Now they are saying some companies that they are going to publish it because it's a patent, right? They don't want to say what they are doing because it's a trade secret. But yes, it is being done in some developed countries. Then the second kind here is the stamen-less male sterility. It produces flowers with only a vestigial development of the stamen, which under most conditions produces the, it will not produce the pollen. So that is, the stamenless. Then the third one is the functional male sterility I was talking about, the mutants that includes the positional sterile. The genes are there, specific genes and genotypes are identified. PS and positional sterile 2, PS2, and clistogamous 2, that is CL2, and dialytic DL, and exerted stigma EX. This with the specific genes they have identified. Okay, these are functional male sterile. Of all these, the most studied is the PS2 gene. Right, the PS2 gene, the testing of PS2 line under the large range of environmental conditions showed that occasionally the retention of the pollen was not totally consistent. However, further studies have shown that it is possible to improve the efficacy in the use of this type of sterility, even in case of emasculation. Okay, sometimes, if it is not there, you can do the emasculation. It can be conducted at anthesis, which is significantly easier. That means it's not full proof, but yes, this can be used. So PS2 sterile parents have been used in few countries like Czech Republic and Moldova, Poland, Bulgaria, and a number of hybrids possessing functional male sterile seeds parent have been released there, right? How they are doing it, all the problems, you got this, that they have to resolve. And they are commercially doing means with that only they are, existing, or they are making use of this genetic tool. The exerted stigma, this the style is grown extra, the stigma is coming out of the anther cone in tomato. Anther cone, it is there, right? So once it comes out, it is not amenable for the immediate pollen whatever it is released from the anther cone, right? So it's not associated with the alterations in the anthers, as their dehiscence is normal and they produce the viable pollen. So that functional male sterility, what you said, it is anther is there. It's the only thing is pollen. I mean, the anther is not released. I mean, open and pollen is not released. But here, everything is there. Everything happens. But only the position, that the stigma is above that anther cone, and it is not amenable for the natural pollen transfer because it is a highly self-pollinated crop. Then you can have the one more line, and then you can go for the hand pollination. It is due to the excessive length of the style which protrudes from the cone and holding the stigma out and away from the concentration of the pollen in the central space, and which effectively produces the functional male sterile phenotype. This is quite long back only it is being reported, but used of late. However, its expression depends on the length of both style and anther. These lengths being polygenically controlled and varying significantly depending on mainly on the temperature. Again, you look at here, the temperature, it's adaptation by the genotype. Sometimes they, the stigma is getting exerted because of the high temperature. That means it is restricted. So for growing the, producing the hybrid seeds, which season you have to do it. So the most, all these are there, but the most used genes for producing the hybrid seeds in tomato are MS series and the functional male sterility controlled by the positional sterility 2 gene. So only these two are commercially exploited. Other, even exerted stigma and all, the success, we are not getting it or reported. In spite of the significant amount of research on the application of genic male sterility in tomato breeding, up to now, male sterile lines have not been used on large scale in tomato hybrid seed. Although few seed companies, and whether what technique also they are using, you may not be knowing. But yes, it may be a trade secret. We don't know. But by and large, it is said, still those developed countries, they send the parental seed material to the countries where the labor is cheaper, and they are producing the hybrid seeds. Okay, that is the hiccup. That is in case of tomato. Okay, just a minute. Yeah. So that is the story of tomato, but we are producing by hand emasculation and pollination only. It is possible. Some of the seed companies are using it. The next crop is the pepper. Just a minute, please. Okay, the next crop is the pepper. Much of the commercial seed industry F1 hybrid ಇದು ರಮೇಶ್ ಕೊಟ್ಟು ಇದೊಂದು ಮೆಸೇಜ್ ಬಂತು ಅಂತ ಇದನ್ನು ಆಫ್ ಮಾಡ್ತಾರೆ. Okay, much of the commercial seed industry produces the F1 hybrid seeds. The reason with extremely cheap labor costs such as China and Thailand. You are doing it right. This has led to the expensive seed for this pepper cultivars because all this you are doing it manually. So here the genic meal sterility is being employed to produce the few hot pepper hybrid cultivars. After roguing the fertile plants from the seed parent, pollen may either be transferred by humans or by bees. The first public sector hybrid CH1 was developed at PAU, Ludhiana, using this MS12 genic male sterility. This male sterile line has been developed by utilizing the male sterile line introduced from France, at PAU. So the same technique, of course, at IHR, they have identified 3 cytoplasmic and genic male sterile lines, and they are using it. Now, actually, they got it from the Korean hybrids and, of course, the local sources also. So they are using the CGMS system. At IHR CGMS system, they are using that. They release the hybrids and they are producing the seeds. At PAU, the genic male sterility is used. But I tell you, the CH1 and CH3 hybrids, they are released. They're marketed with some private seed company hooking up and all that. But later they encountered a lot of problem because a lot of skills involved in identifying the fertile plants in hybrid seed production plot. That is a very risky, and later it has become a problem. I think PAU, they know that what is the problem here, because at farmers level you cannot expect that much of skill and identification before the pollen is being dispersal. So that's why it is not I would say that's successful. But still, they have some years. Definitely, they have marketed this hybrid CH1 and CH3. Seedlings should be transplanted at the planting ratio of 2:1. That is MS12 into that parent line. Also, it is given here. That is the Ludhiana local selection, and then CH3 it is MS12. Again, the same female parent, male sterile parent they have used, and S2530 is the male parent, and they have done the business for some period of time, but with some problems also. So this is what they actually at PAU they have used this and the male sterility sources and employed it for seed production. Right? How they have done also it is then. I think CH3. This is what the system. How the identification of male sterile and the male fertile plants in the seed production plot before pollen shed. It is difficult, and you need to go for some markers only. That is the way how one can look at. And of course, at IHR, this Arca Sveta, it's F1 hybrid CGMS system they have used and release these hybrids, and they are producing the seeds, as you heard yesterday from Dr. Nagavoda, technical officer, that they are doing it. And Arca Meghana is one more hybrid using CGMS system. Dr. Madhavi Reddy. Arca Harita is one more hybrid they have developed by using this CGMS system, and they are producing the seeds, hybrid seeds, by using this system, and they are selling the seeds also. But yes, I told you there is a problem of identification of 50% heterozygous and the homozygous recessive in hybrid seed production plot. You need to go for use of the markers. So development of molecular markers also being looked at PAU. AFLP marker tightly linked to the male gene MS1. It is reported AFLP marker tightly linked to the male sterile gene MS3. Lee and co-workers 2010, they reported molecular markers linked to the male cell gene of MS509. They could not find out. Maybe with certain effort, maybe new one they can get in. So from 1996, AVRDC received funding from Semini seed companies for CGMS alliance development. Source material donated by Korean seed company, Chungang seed company. So each year, 10 to 15 chili lines test cross to Korean CGMS lines to determine whether they are maintainer B lines or restorer C lines. And the 6 B lines were backcrossed to the MS F1 to begin the process of A line conversion. 5 to 6 backcrosses needed to convert the B line into A lines. And this is what the effort they are doing it. That means many times the AVRDC is a good source for getting the parental seed materials. And of course, then how you can convert this maintainer B line, cytoplasmic male sterile A line. Also, you can place this male sterility in different background. You can place it. So using AVRDC material, they have developed CGMS lines. Now you can develop your own. Once you have got that CGMS source, you can introduce this CGMS system into different genetic background, and then you have opportunity for exploiting newer and newer hybrids. So with the good combiners, if you have got lines identified, you can transfer this CGMS system, and you can look for the restorer gene also for that particular CGMS line, and you can develop the hybrids. That is being done in some of the institutes. So this one more thing I just told you that this CH1 and CH3 they encounter the problem of not identifying the fertile plants, and then that is contributing the self seeds by not identifying in a hybrid seed production plot. So here one more the gene, actually the hybrid seed production based on CMS combination with the lethal gene and the female sterile pollinizer. That means the lethal gene allele is being used. It is introduced in the female parent, and if selfing happens there, then the non-hybrid plants will die due to the action of the lethal gene, and they are going to automatically you are resolving this problem. How much that is a success? It's okay concept. Maybe some of the seed companies have successfully being used this technique, no doubt in that. But this can resolve the issue of genic male sterility, where 50% segregation is there in the seed production plots. Identification of the fertile plant you can resolve by this auxotropes or nutritional mutants or lethal genes introduction into the CGMS system. Molecular markers for CGMS line, development of CM specific scar markers to distinguish N cytoplasma from S cytoplasma. That also they are looked at, and develop the CAPS marker linked to the PR locus related to the partial restoration of the fertility, because a lot of markers you have to use it for identification and of this transferred gene into the newer parental material. It's always there, and of course these markers also can be useful in hybrid seed production process also. So that is the story of capsicum annum, and a lot of papers also there in capsicum as such. Although capsicum and chili belongs to the same species in genera, but they are the fruit size is big, more number of seeds are there. Still, people go for hybrid seed production by manual in protected cultivation, just like you do it outside for the tomato. But there are systems, CGMS system reported. But the thing is, stability is the problem. Maybe some of the seed companies are using it. They may not publish it as a trade secret. The next crop is the genic male sterility. Genic male sterility is found to be controlled by 2 recessive genes, which was identified at Indian Institute of Horticulture Research, Bangalore. And this is what the Arca Nikita they have developed. And this is the male sterile line they have used. GMS4. This Arca Nikita having the parents GMS4 into IHR 299, what that male parent. But yes, here also identification of this 50% segregation material is there. The breeder himself, or many times it is not possible if you are producing the seeds in large quantity, but it may it's the same problem is what it is being faced in chili. Same thing you can also get in okra, but it has to be done because the the only difference is that number of flowers or the process of the continuous flowering is more in case of chili compared to the okra. But here also that 50% segregation issue one has to look for. Then the muskmelon genic male cell system is being used at PAU. MS1 to MS5 genes have been identified. Several male cell lines are there. So MS1 used in muskmelon hybrid seed production governed by single recessive gene at your in India. In hybrid seed production, female and male are grown at a ratio of 4:1, right? So PAU that they have produced this hybrid, right, using the MS1 female parent. Same is the case, that 50% segregation. So here in cucurbits, okay, you can do it because the seed requirement is less and the flower size is big, more seeds are there per fruit, it can be managed. So use of Gynoecious sex form. Yes, Gynoecious sex form has been commercially exploited for hybrid seed production in muskmelon at PAU. The female line, the Gynoecious line, is maintained by inducing the staminate flowers by spraying silver nitrate. Of course, there's the other way you can maintain it. You induce the maleness to maintain the Gynoecious line. If the parental line is andromonoecious, the perfect flowers must be emasculated. So andromonoecious means, right, you have got a male and also you got this hermaphrodite flowers. If the female parental line is monoecious, hand pollination can be performed without emasculation in insect-proof conditions. You can do it. So alternatively, spraying of ethereal or on monoecious line suppresses temporarily the male flowers, and the bees can assist the pollination. Again, here you have to standardize. It's not simple. What stage it has to be applied and what chemical, what concentration, whether it responds or not. All these are the issues that are going to be specific to the line. So 5 recessive genes of male sterility also have been identified. As I said, it is being commercially used in muskmelon. Then watermelon, that is, the seedless watermelon production, tetra 1 and tetra 2, tetra 3 and tetra 4, the tetraploids produce. They are used for commercial production of seeds of seedless watermelons. Tetra 2 line has been also used at IRI for production of seedless watermelon hybrid, Pusa Beda. You know this story. Of course, many seed companies have come out with that, and it is only the sex forms are being used for seed production. The cucumber hybrid seed production is done either in a greenhouse or a field environment by hand or insect pollination. For large-scale commercial use, the open field with the strategic placement of rows such that the cross-pollination can occur between the lines of opposite sex types. So typically 1 or 2 male rows are alternated with 4 to 5 female rows from which hybrid seed is harvested. And again, this range it is given because it depends on the kind of parent you are using. It may vary. So hybrids commonly result from Gynoecious into Gynoecious, or Gynoecious into Monoecious, Monoecious into Monoecious, and the Gynoecious into hermaphrodite lines mating. Anything you do it, maintenance is possible because you have chemicals being used to produce the male flowers. Okay, you can maintain, but the genetic system is also being looked at. For that, genetic information, I think I don't explain you. There are 3 genes. It is very complex. Genetics is there. And I just want to say it's one important point. ACR, okay, totally good. Earlier Smith reported genetics of sex expression in cucumber. So later it is clarified by using these 3 genes only. Just symbols are changed. ACR, the Gynoecious it uses. ACR is partially dominant to ACR. So that is a hiccup here. Okay, this genetics. Okay, if you know it's breeder, you should know it, right? But what is that seed producer should know? So unstable sex expression is the problem in Gynoecious lines, because the Gynoecious into hermaphrodite, if you use, what is going to happen? The F1, it is going to be the ACR is dominant in both hermaphrodite sex form, right? The hybrid is going to be ACR dominant. So Gynoecious is more stable. That means the maintenance is going to be easy there. If you cross the Gynoecious with the Monoecious, the ACR is dominant in Gynoecious, whereas in Monoecious it is recessive. Your progeny is going to be heterozygous. So it is said it is incomplete dominance. That means it's not very perfect for the female flower production. For that reason, maintenance one should use hermaphrodite lines rather than the monoecious lines. Then only your hybrid seed production plot will be little bit free from, as cucurbits are very, very, the unstable with the sex expression. If your genetic system is also, partial dominance, means it will add to the problem. So for that reason, you had to go for Gynoecious into hermaphrodite kind of maintenance. Therefore, use hermaphrodite lines for maintenance. And of course, the hybrid seed production, So cultivation at high density under high temperature and long days strongly promotes. So now you have to assess these Gynoecious lines, whether they are very stable or not. For that, you have to give the unfavorable condition for the Gynoecious lines. That means the female expression, it is the short day and low temperature is good. But you expose these lines in a very high temperature, long days, then you can assess the stability. Otherwise, it is going to be a problem for the seed producer. So for achieving the stability of Gynoecious, hybrids need to constitute hybrids from Gynoecious and hermaphrodite inbreds as a female and male parents respectively, and not by Gynoecious into Monoecious. That is the way, the importance the seed producers should understand. If you got a hybrids given by using the male parent as a maintainer, the Monoecious as the maintainer, then you have a problem at your hand. So if it is having, being maintained by hermaphrodite, then your problems are a bit resolved because of the instability of the Gynoecious line, which may suddenly produce the male flowers. The same thing is being, Dr. Dhananjay, so whether the next speaker, because the many crops are there, I'll just go through. How much time is there? Yeah, how much time is there?
Sir, you can take another 15 to 20 minutes, sir. No issue.
Okay, 15 to 20 minutes. Okay. So use of pumpkin, the Gynomonoecious, right? Gynomonoecious and Monoecious lines are planted alternatively here as the system is available, and you can go for hybrid seed production. Whatever it is being explained earlier, also same thing is applied. But the genic male sterility is also looking, because of this unstable sex expression, people are looking for male sterility system here also. It is why the question can ask somebody, why, sir? It is already, that system is available. Why not? They are trying to explore the other genetic tools so that whether the problem, what is there in the other system can be resolved, and which is more comfortable, which is less prone from the problems they are looking at. That's why it is being explored other genetic tools also. Genic male sterility is also looked at here. So that causes the male flowers about has been described in Cucurbita pepo, all these pumpkin species, Cucurbita pepo, Cucurbita maxima, and Cucurbita moschata. So the genes like MS1, MS2, and MS3 have been identified and explained. However, it may be difficult and expensive to maintain male sterile line populations, as it is said, it's explained that, right? So it is not like, you cannot think like, the rice, or the sunflower or the other field crops like importance you can get in for these vegetable crops. So pumpkin, it's a very minor crop. Vegetable as such, you don't find individual, crop institute. That's why, little, scientific manpower is there, and effort is also going to be, the diluted. That's why you can't express unless the crop is very, very, commercially important. Onion, yes, it is commercially important, and a lot of, efforts are being done here. And almost all the developed countries, more than 90% of the area hybrids are occupied in onion. The first CMS source discovered is nowadays. Okay, so here one thing we should know, that the CMS system is being used here, but the CMS system itself, you have got 2-3 types are there. CMS S and CMS T. Because until recent past, people were thinking that we are handling the same thing, but later they, by trying to find out the restorer, they could be able to say that, oh, this is a different kind of male sterility system, and they named it as a CMS or T, right? By when in there. So, Seves, 1973, analyzed the genetic basis of this type of CMS and showed that the male sterility in this case was restored by dominant allele at a locus A, or a dominant allele at 2 complementary functioning loci, the so-called B and C loci. So by this only they could able to say that, oh, this is a different kind of male sterility system. I mean, the CMS gene is there. Currently, both types of CMS are used in commercial hybrid seed production in onion. All the CMS source is probably used most frequently worldwide, as only in Dutch and Japanese cultivar CMS was discovered. And availability is also there. Question. D. Corsell and co-workers, they showed them via molecular marker analysis that there are two groups of cytoplasmic male sterility system, namely S and M group. Look at here. So further they are explained. Otherwise, we are thinking only one system. Now, what they said, in M group, there are two subgroups. Those are N and T. Now, popularly, T and S only we know. So totally, actually, two groups are there. One is S and another is M. And within M, you have two types. One is N and another is T. Okay, the N and T. So within these groups, M groups, variability was found, which makes the development of molecular markers which can distinguish both groups very difficult. Variability is there. It's not breeding true that it's throwing the variability. Molecular marker finding out through only molecular markers they could be able to identify that, oh, these are the two different types are there and all that. But within this M group, you have got variability and it becomes difficult to identify them. So, however, in the S cytoplasma group, no or very little variation was discovered, and which has led to the development of PCR markers that can make, you can easily distinguish this. So that's why this S system is more frequently used than the T system. T system is being used in a few, that's why it is restricted, I told you. It is restricted in few of the countries and few of the seed companies. And it has got some kind of advantage. Also, it is thermal, not stable. It can respond to the change in the temperature. That becomes sometimes advantage. You can alter, to just suddenly you can break the mesh solid and or can be used for maintaining the that lines, the parental lines. So. So here, one more thing is also there. The PCR markers that distinguish between the S and other two types of cytoplasmic male sterility. It is possible. Now at least S you can easily identify, but another group, if it is not there, so to distinguish between N and T, it is difficult. So yes, a lot of work has been done in this. Harvey in 2000 proposed that S cytoplasma was introduced into onion via pran, that Kashmir variety, a viviparous triploid onion that actually source of this S cytoplasma, and that N and T cytoplasma originated from the progenitor of the M cytoplasma group. So he proposed that this Arnium vavilovi might be this progenitor for that. So microsporogenesis in both CMS sources takes place in a different way. Actually, yes, it is normal, whereas in case of M group, it is, or even T group, it is abnormal. Meiosis is different. The frequency of the restorer allele for S can also differ among the populations. So you have to look for. So especially in restorer means in cytoplasmic restoration also, you have to look for maintenance. And especially in tropical populations where the frequency of M allele is close to 100%, it can become a problem for the discovery of maintenance, which are of pivotal importance for the development of F1 hybrid seed program. So this is the problem. So it is M allele is, it's almost it is difficult to get restored. That means getting the fertility there. Of course, may not be in hybrid seed production, at least for maintenance you need to. So the restorer locus of CMS has been located on chromosome 2. So a tightly linked molecular marker to this restorer locus, which can facilitate the breeding process of new onion cultivars. That means you want to transfer into a new background. So these are some of the issues of onion male sterility, that is S and the T cytoplasma type of male sterility, and looking at the restoration also in the nucleus. That is, the entire system. You want to make it foolproof and easily identifiable. Now, what is that hybrid seed production? It is, of course, hybrid seed production. It is, the global temperate onion seed market consists of 90% F1 hybrids. However, in tropical, the frequency of OP varieties is still around 50 to 60%. So that means it is still there. The hybrids, that is the reason why the hybrids are not popular in onion. It's only popular in other countries, the developed countries, or the temperate countries. It is there. So here, the exploitation of heterosis is one aspect which is advantageous, and also increase the uniformity. So uniformity is one more thing. We are looking from the hybrids. So that is less evident in case of onion. The uniformity is less evident. What is the reason for that? These to develop the lines, if you go for more number of selfing generations, the inbreeding depression is coming in the way. What is that? Normally they do it one or few generations of selfing and immediately maintain it by massing. So variability is there in the parental line itself. Then naturally you don't get the, if parents are a little bit variable, then the hybrid is not going to be uniform. So that's why you cannot say that uniformity is there, more uniformity like another crops you are saying. So if you, that's why so uniformity is not there. And the heterosis, you said, the yield increment. Whatever size of the onion you get it, by spacing adjustment you can get the yield. So what is the advantage? In other crops you have got more branching, more fruits, big size fruit and that and all, or you are saying that you are going to get the heterosis. But here it's a single bulb is there, and of course the size also, if the size is smaller, you can go for closer spacing and you can get the same kind of yield. So hybrids are not offering even the higher yield, neither the uniformity also. And of course, one more thing in developing countries like India, although you have got more the sized shaped bulbs are there, you can sort it out and you can sell even the smaller bulbs in the market with not much big difference. The optimum size may be marketed for the 30 rupees a kg, and the discarded small size you can sell it at 20 rupees a kg. Whenever scarcity is available. I mean, the scarcity is there in onion, definitely. So that means all these things are. In addition to that, the hybrid seed cost is also going to be high. It is only the private seed companies, because their the technology is protected, they come into, right? The technology is protected, meaning that every time farmer has to go for buying the seeds. That's why they try to push this technology in onion, although it is not that advantageous as in terms of, neither in terms of yield nor in terms of uniformity, and of course nor in terms of the cost of the seed. But they can protect the technology. They are, but still we have got some hybrids here. Maybe the hybrids in terms of the survivability of the seedlings, it is the combination of the genes that can offer. That is very important. What is the advantage? Actually, it is the dispersion of partially dominant genes. Dispersion of partially dominant genes can be combined easily, because either of the parents have got dominant genes, that will come to the hybrid. That is the one major advantage, disease resistance or anything. So that can be exploited here, and only that is the only factor which can promote the hybrids in onion. And at IHR they have developed this hybrids. They are marketed. Of course, these hybrids. I don't know how much they are marketing and all that, but definitely not much the business, because the cost is going to be more. Whatever it is, the Arka Kirtiman and Arka Lalima are the two hybrids from IHR. They have been developed and commercially also cultivated. They are good hybrids, no doubt, compared to the OP, right? But the thing is the cost and availability of the seeds. The private people, they come with the advantage, profit margin only. If you ask them to put the same, the price, it may not be there. So availability of the seed is also going to be the factor. Okay, that is the story. Then then the carrot. Okay, here. Okay, time is carrot. You have got Male sterile lines are there, right? That is the CGMS system, cytoplasmic genic male sterility system is being used. Here also, of course, cytoplasmic male sterility is sufficient, but whatever it is, there's. It is, you know, the popularly petalloid and brown anther type of male sterile lines are available, and male sterile lines are produced white to green colored flowers, right? While the fertile, they one produce the white flowers. You can easily identify these male sterile lines, and they are used for hybrid seed production. The male sterile and the pollinator lines are grown in alternate rows of 4:1 or 8:1, and the hybrid F1 hybrid seed harvest from the female line. So male and female parents should be having the synchronized flowering for better pollination, that you have to ensure while identifying the parents. 5 to 6 bee colonies, even in onion also, beneficial for affecting the successful pollination. So in carrot, 3-way single cross hybrids are made using the male sterile lines as one of the parent. So this is being done. And of course, the cabbage and all, it is a self-incompatibility system. I told you have to construct the self-incompatible lines, 2 lines, differing the 2 parents, parent 1 and parent 2, differing in the self-incompatible alleles. That is being, you know, the task of the breeder. The maintenance of that, the self-incompatible line is the bud pollination at that time before the flower opening. If you open the flower and pollinate it, the pollination happens and the seed set will be there. But if you naturally allow it, the time the flower opens, the stigma becomes receptive, the incompatibility operates, and the self-pollen will not be able to germinate onto the stigmatic surface. That happens, but the breaking down of that is also reported. That's why people are looking at some other alternative methods. So for maintenance, even they go for bud pollination 1 and carbon dioxide concentration increase in the polyhouse, or 3 to 5% sodium chloride treatment also can get you the pollen in the self-incompatible being germinated, and you will get the sub seeds. That's maintenance, you can do it. So hybrid seed production is quite, you know, you can harvest the seeds from both the parent. That is one advantage here, right? It depends on, you know, if you have got, if any cytoplasmic gene difference other than this self-incompatibility, if it is there, then only you can restrict to the one parent. Otherwise, if both these lines, I mean the parental lines are equally good, definitely you can collect the seeds from both the parents. Nuclear genes equal contribution is there, no problem. If cytoplasmic difference other than the self-incompatibility is there, then you can restrict to the collection of the seeds from the one parent. Otherwise, most of the time it is both the parents you are collecting the seeds, and seed yield is going to be better. The only problem here is the stability, right? So commercially they are producing in cabbage and cauliflower. So genic male sterility they are looking at. Cytoplasmic male sterility, of course, it is being transferred from other brassica species, Brassica oleracea, from the radish, and later into the cabbage by repeated backcross. It has been done. And you know that process is on. Maybe some seed companies are ahead in exploiting this particular kind of male sterility. So same even in cauliflower, because both these are the same species, even species also same, only botanical variety difference is there, is easier. Other brassica species also being used for transfer of this genic male sterility system. Otherwise, self-incompatibility is the only problem is that stability of that. You know that even genetic constitution is also given as to how this being used. I shown you at the beginning only the 2-way, 3-way cross hybrids. So production of hybrid seeds, you can just go for the placing these parents and maintaining the honeybee hives. You can produce the seeds. You can collect the seeds from both the parents without much difficulty. Okay, so. Yeah, you can produce the hybrid seeds by single cross, double cross. That S allele you have to construct in the parents of different alleles. So the female and male parent ratio and population on a hybrid seed yield in cauliflower, you know, just some data, you know, ratios. It depends on the particular line also you are using. So female 2 line and the male 1 line, always the yield is higher. The population is also, because you are going to collect. You know, if you are collecting from the both the parents, it may not be an issue. You can have equal ratio. But if you are collecting from only one parent, then there is an issue and there will be a reduction in the yield. So India is the second largest user of hybrid tomato seeds after USA. Cabbage and tomato have largest areas under hybrids among, more than, like, I think now it's almost 50% areas being covered by these hybrids, even more than that also. Somebody can survey and get it. These two crops are very successful as the hybrid introduction is there. The main states in which the hybrid seed production is being carried out: Karnataka, Andhra Pradesh, Tamil Nadu, Maharashtra, and tropical carrots. Tropical carrot, radish, cauliflower is being produced. Seeds are produced in Punjab, Haryana, UP. Temperate vegetable seeds being produced in Jammu and Kashmir, Himachal Pradesh, Uttarakhand, and it's just hybrid seed usage in tomato and chilies in Southeast Asia, some of the countries also, and how much is required. You can just imagine the volume of seed requirement. It can be still more if area is expanded. So I think what I wanted to say, but within the time, I think I explained, and many things. If any questions are there, we can discuss. Yeah, Dr. Dhananjay.
Thank you so much, sir, for presenting such a elaborative and in-depth information. Now, with the permission of speaker, session is open for discussion. If any questions are there from participant side, means you can unmute yourself and you can ask the questions.
Good morning, Mulge.
Yeah, good morning, sir.
This is regarding, again, I am Hegde, representing the Nunam seeds.
Okay.
Maybe IP or the varietal protection is very, very important for a company like us. Maybe yes. My question is whether you have any idea or the updates on the pollen testing part as far as the solanum is concerned. Maybe we know only the acetocarmine or the growing in the agar or something. Any new advanced technology of pollen testing or pollen viability impact on the seed quality?
Yeah, the pollen testing, I think the first speaker, Dr. Rajshekaran, and his lab itself is a good the source of information, also facilities at IIHR. Dr. Viti Hegde. Hello.
Yes, sir. Yes, sir.
Yeah, you can directly consult that Rajshekaran, because okay. At the time of my PhD also, I did this pollen germination studies. Actually, we were trying to look at for assessing the combining ability, building any relation between the pollen vigor and the heterosis. But that time, that Alexander Stein and other things were there. Now also it is there. I think for this particular question, I think you can directly contact with Dr. Rajshekaran. Dr. Sure. Lab is the ahead in developments.
Sure. Thank you, Mulge.
Okay. Yeah.
Sir, good afternoon, sir.
Good afternoon.
Sir, myself JP Khatod from Dr. PDKV, sir.
Right.
Sir, I want to know the effect of pollen on the stigma with respect to the metaxenian phenomena, in which the cucumbers nowadays in summer are very bitter. So, any solution from genetics point of view?
Thing is that genia metagenia effect. You are referring to the bitterness, isn't it?
Yeah. Hello.
Hello. Can you hear me?
Yeah, sir.
Yeah. Bitterness, I tell you, the effect of what is that? Most of the time, what the germplasm is available with you, that is being not purified. One thing, and the second thing, this the genia and metagenia effect, definitely, that particular genotype, you can identify and have the the line for having free from bitterness. But at the time of seed production, somewhere some line is there and it is not pure with that bitterness gene, and if the pollen is falling, only those seeds, if you plant it, definitely you will get the bitterness, right? It is the pollen, it can come 1,500 meters, it can come by surrounding areas. You don't have the control. Okay.
Okay, sir.
The complexity is that, okay,
Yeah.
you have maintained your genotype, right? But
Yeah,
surrounding area, you cannot say that from where the pollen has come, and suddenly your one fruit is becoming bitter,
Yeah.
isn't it?
Yeah. Yeah.
Yeah, that is the complexity of the problem. Although you are maintaining, of course, if you are doing it in case of the seed production or anything in the protected condition, and you are not allowing any pollen, definitely you should not get that. Your genetic material is pure; it is free from that gene of bitterness, and you are doing it with the complete the screening with your structures. Definitely, you should not get. Otherwise, outside you don't have the control.
Okay. Means we have to take in protected.
Pardon me. Hello.
Yeah. Yes, sir. Means we have to do this seed production in protected way.
If you do it, but many times practically it may not be feasible.
Yeah.
Practically it may not be feasible. Right, that control factor is the issue. Leave apart your seed production, even commercial production also. Your seed may be free from bitterness, but suddenly from some other wild types, any pollen comes, or some other genotype where it is not completely free from that gene. If plants are there, if they are contributing pollen, definitely those fruits which have got that pollen, you are going to get the bitterness.
Yeah. Yeah.
Isn't it?
Yes, sir.
Yeah.
Thank you, sir.
That's the only way you can. Okay.
Hello, sir. Hello,
Hello.
sir. I have one question, sir.
Okay.
Hello.
Okay. Yeah, please.
Hello, sir. Sir, my question is whether the CMS lines are can break under certain environmental conditions like high temperatures.
Definitely. That's what I told you. In the capsicum annum, the CMS, it is not stable. Suddenly it breaks. That's why they are using the oxotropes. Even not just any kind of, not just genic. CGMS system itself, it was not stable. And I tell you that CGMS system in capsicum annum, what they studied earlier, the fag and the flowers will become the fertile. It breaks. The stability is not there. That's why don't collect the fruits from the later stage. And if the temperature suddenly raises, breaks. So these problems are there. They are not stable. That's why people they construct the markers. The axotrope markers, why they are I explained you. It is only to avoid that if selfing happens because of that instability, automatically that anthocyanin-less mutant in tomato decided they are having this. Of course, that is used for that, but this is for instability of the male sterility also you can resolve. That is known. Genic male sterility, 50% segregation, you are using this marker. Same marker, oxotropes are anthocyanin-less mutants decided. If it is constructed, used along with the CGMS system or your CMS system, if selfing happens because of the breaking down, that the next screening is by this marker, right? If it is anthocyanin-less, automatically the seedling stage itself, that seedling will die. It germinates and it dies because no chlorophyll synthesis mechanism is there, and that will die. But maintenance, I told you again, you have to spray, you have to develop your protocol, standardize. That's spraying that thiamine or what some amino acids that genotype should be able to
Sir. Thank you, sir.
life cycle and get the seeds. Okay, right.
Hello.
Yeah. Okay. So if no questions are there, Dr. Dhananjay, I think the next speaker is also must be.
Sir, Satvir Roti, sir, is.
Yeah, please.
Sir,
Please,
I'm Satvir Roti, actually from Netra Seeds.
Yeah, I know.
I'm not a breeder, but I have some question for you, sir.
Yes.
Sir, actually, you were said in the okra, some GMS, this one breeding. So we are facing some color problem of the okra fruits. Actually, there's the bottom of the fruits always getting yellow, most of the habits. If we dark color more, we made a dark color and the disease tolerance will be come down. If disease tolerance is increasing and automatically food quality is coming down.
Yes.
So any other new techniques and some things we can help us to breed, because I can convey that message to our breeder.
Actually, it requires a lot of, the efforts. What you say, we are getting the quality, the lighter green, the fruit color. It is all right. Your disease resistance is associated. Some of these are always a problem, right? So it is only you need to have a backcross with the disease resistant genotype with the dark green fruits. So several backcross generations can resolve your issues. You have to put that disease resistance into the dark green fruited accessions. That requires 4 or 5 backcross generations to. First you have to have disease resistance. You said it is going down, and it's not like subject. It's yes, disease resistance is going down means it is linked. Again, the kind of genetics is involved in that particular disease resistance. Polygenic, it becomes very difficult, right? What kind of accession you are having? If it is single gene, definitely you can easily transfer this disease resistance into a dark fruited genotypes. It is possible. You can break. Even they are tightly linked also. You have to put on certain techniques like interseminating and all. The professional breeders, they know all this, but it requires time. It requires time, number of generations.
Okay, sir. Thank you, sir. Thank you so much.
Okay.
Thank you so much, sir.
Okay.
If there are no questions from participants, means we will wind up this session at this time. I extend my thanks to Dr. Ravindra Mulg sir, our beloved dean, for shedding lights on from basic information to molecular up to molecular level with respect to the genetic tools involved in hybrid seed production. Thank you so much, sir.
Thank you, Dhananjay. Thank you.
Now we have another speaker with us, that is Dr. Sridhar Guttam sir, who is senior scientist, ICAR IIHR Bangalore, who is going to deliver a lecture on application of plant growth regulators in seed production. On behalf of University of Horticulture Sciences, Bagalkot and College of Horticulture, Bidar, I welcome you, sir. May I invite Dr. Ashok Suramsi sir to give a brief introduction about Dr. Sridhar Guttam sir. Over to you, sir.
Okay, thank you, Dr. Dhananjay. A very good afternoon to all the participants. I'm very happy to introduce today's speaker, Dr. Sridhar Guttam. He's my best friend also. He is working as a senior scientist in plant physiology, ICAR, ASRP on fruits at the Indian Institute of Horticulture Research, Bangalore. So coming to his academic qualification, he did his bachelor degree in agriculture at Acharya Ranga Agricultural University, Hyderabad.